morning, Allison.
Morning, Kelly. How are you?
Good. How are you?
I'm all right.
Just going to do the cookie run pickup today.
We just see the darkness having this show.
Yeah. And David and Laura's lunchbox would be great.
Oh yeah, really looking forward to that. Let's do our COVID forms.
Yep.
No close contacts for me.
Nope. I just answered a lot of emails last night.
All the emails.
Nope. Across the board.
All right. Let's check your temperature.
Okay.
.3.
Nice. Write that down. Also 98.3.
Twinsies.
Yeah.
Right. First Zoom of the day.
Yes. Let's hop on in there.
Good morning.
Morning, everyone. Bye, everybody.
Bye.
Hi, I'm Kelly Salmon.
And I'm Allison Michael.
And first of all, we'd like to thank our excellent research students this summer who performed great research in their home labs rather than in our NHAS STEM lab.
As the pandemic prevented us all from gathering together for a research presentation, we created this video offering of our students explaining their remote science research. Please sit back, relax, and enjoy the show.
Hi, my name is Joseph Della Valle. And my name is Molly Della Valle. And today we'll be giving you a behind-the-scenes tour of our home lab and telling you a little bit about our projects. So as we come over here, I would first like to talk about my project. For my project, I'm taking various pictures with infrared filters using these Raspberry Pi cameras. NVI is a tool that we use to measure plant health through photosynthesis. As you can see over here, this is a NDVI image. All the green in this photo measures plant health. Over here you can see my section of the lab, and my project we'll actually see more of later. But basically I am also taking photos with a Raspberry Pi and a camera, except using an active illumination system. Active illumination is basically anytime we're taking photos and adding light to the situation. So for example, the flash on your phone is an active illumination system because when you turn it on and take a photo, you're adding light to the situation that you're taking a picture of. My system is a little bit more complicated than the flash on your phone. It can actually provide active illumination in different colors and from different angles, and you'll see how that works a little bit later also. The last thing I want to show you is our Zoom station over here, which has our laptop, a speaker, and our lab notebooks, which we've been using to take notes throughout the entire camp. And also over here you can see our brother John sitting on the couch. It's a very authentic sighting. He's been sitting there for most of the camp. And now we're going to head downstairs to see the dark room where we took most of our photos. So here we have our dark room, and as you can see, there are no windows in here. So the light is very controlled, and it only comes from sources that we can turn on and off. This makes it perfect for taking photos, especially with active illumination, where I only want the light that I'm providing to be visible. So over here you can see my system is set up, And we have my system here. Here you can see my camera, which is connected to a small computer, the Raspberry Pi. This is an Arduino, another small computer, and it's connected to our ring light here. This is what actually provides the active illumination in my system. Each of these lights can turn on and off all by itself and can change to different colors. So here it goes around the camera, and that's how we get different angles of active illumination coming through our light. Here you can see one of the models I use. This is actually some color filters that are taped to a white piece of paper, and that's a really good model for taking pictures with colored light. And you can just see here that we've got the rest of the connections for my Raspberry Pi, a monitor, a keyboard, and a mouse.
So I'm using basically the same setup, except without the ring light, the Arduino, and my keys just looks a little bit different. I put these filters, this one's an infrared filter, because it's black, in front of the camera and take pictures of a little LED model that I showed you earlier.
Welcome to my basement lab. So this is a P1000 pipette from Eppendorf. These are really nice pipettes. Oh, this looks like we've got some NHAS merchandise. So here is where students perform cutting-edge research and launch STEM careers. That's New Hampshire Academy of Science. Mm-hmm. This summer, I developed a research project to determine if the Mink Brook is affected by small-scale farming. I wanted to investigate two hypotheses. First, does this brook's water quality decline as it flows from its mountain source to its output near downtown Hanover? Second, is the brook's quality affected by the presence of a small farm along its banks? To answer these questions, I set out to sample water at 4 sites for 2 weeks. After collecting my samples, I performed standard water quality tests. I then plated bacterial samples and waited for colorful and interesting colonies to grow. Testing on site was a lot of fun. By the end of 2 weeks, I had ample data to consider my hypotheses. After completing basic testing in the field, I returned home to prepare bacterial plates. During this repetitive task, background music, like the Dvorak you hear now, was my constant companion. Comparing my bacterial plates between sampling sites also helped me consider my hypotheses. Now I was ready to present my results. My data allowed me to confirm both of my hypotheses. I'm looking forward to finding out more about those interesting colonies through PCR in the NHAS lab.
Hi, my name is Laura Shaner. I was in the water quality group for this year's NHAS summer session. For my project, I examined the water quality of two different parts of Mascoma River. Mascoma River flows into and out of Mascoma Lake. For my project, I examined two sites. The first was the water that flows into Mascoma Lake, and the second was the water that flows out of Mascoma Lake. I compared these two sites to determine whether human activity within the lake negatively affected the water that comes out of the lake. I hypothesized that the human activity within the lake did negatively affect the water that came out of the lake. I also hypothesized that it affected the bacteria negatively and the macroinvertebrates. My materials for this project included various ChemEts tests, including phosphate, ammonia, and dissolved oxygen. Also various other tests including pH, turbidity, and temperature. I also collected bacteria and macroinvertebrates. For macroinvertebrates, I wanted to see if there were pollution sensitive or pollution tolerant macroinvertebrates, which would indicate better or worse water quality. My results from this experiment were pretty similar for each sites. I concluded that the water quality was pretty good. All of the tests came back with good results. They were pretty similar for each site, and although there were variations, even though there were variations, they still remained within the safe levels of fresh water. For my macroinvertebrate collection results, I collected similar macroinvertebrates, mostly stoneflies and mayflies, which are pollution sensitive and indicate good water quality. My bacteria collection results were the only results that raised a bit of a red flag, although those need further investigation in the academic year. Thank you.
Hi, I'm Caden Vandola, and I tested edamame using PCR. On the left is PCR, and on the right is gel electrophoresis. Here we have TB buffer and the pipette tips. These are the pipettes. On the left is the scale, and on the right is the vortex. And the centrifuges. The first edamame is this Cascadian Farm non-GMO verified. The second edamame is organic edamame. The third is Sea Point Farms non-GMO verified edamame. And the fourth edamame is Steamable's non-GMO verified shelled edamame. My hypothesis is that edamame farmers sell genetically modified products as non-GMO. In the first PCR test, all edamame tested positive for GMOs. For the second test, all edamame tested negative for GMOs. And in the third test, all edamame tested positive for GMOs. And the fourth test confirms that all of my samples did have GMOs in them. These results show that it is very difficult to find a non-GMO edamame. and because of these results, I was not able to complete my original research plan. I would like to thank Dr. Kelly Salmon, almost Dr. Allison Michael, Dr. Peter Feletra, Alexander Kish, the NHAS lab, Kimmel Union Academy, and my parents.
My project this summer was using DNA sequencing to analyze genetic diversity in Cypripedium reginae, or the showy lady slippers. To prepare the DNA to be sent for sequencing, it is first run through the Mini-8 thermal cycler. This amplifies a select region of the DNA that is marked by the microsatellite primers. The PCR samples are then run in the blue gel machine to see if the PCR was successful. The samples are run on gels made of agarose, TBE buffer, and gel green. If there are bands on the gel, the DNA gets cleaned up and sent for sequencing. We then get chromatograms back. The chromatograms are then compared with the sequence from the catalog to find mutations. A mutation could be a double peak in the chromatogram, or that a nucleotide in the sequence from my samples is different from a nucleotide in the catalog.
Hi everyone, my name is Esme Kimber, and I'm an incoming freshman at St. Johnsbury Academy. The project I conducted this summer was on the effects CBD oil has on serotonin levels in C. elegans. CBD oil, or cannabidiol oil, is a cannabinoid extracted from the hemp plant that's been known to treat anxiety, treat depression, and also help ease pain. However, there's a lot of controversy as to whether it actually works. CBD is FDA approved to treat 2 severe seizure disorders. These seizures occur with an increase in serotonin levels in the body. Serotonin is a chemical produced by your body that regulates sleep, your mental health, and other neurological processes. Because CBD is known for treating seizures, and seizures are caused by an elevation in serotonin, I hypothesized that CBD would decrease serotonin levels. I conducted my experiment using the worm C. elegans. These worms are very small, about 1 millimeter, and they're also easy to care for. I used these worms because I have prior experience with them, and they're also very similar to a human's body. These worms also have very obvious responses to serotonin. For example, they'll become paralyzed and stick-like when exposed to too much serotonin. For my methods, I exposed these worms to CBD oil and then exposed them to 3 different concentrations of serotonin to see if there was a dose response occurring. I collected my data by counting the number of worms paralyzed and unparalyzed. For my results, I'm actually still in the process of collecting data, but so far, statistically speaking, there appears to be a significant difference in both of my higher concentrations, but not so much in my lower concentrations. So far, my data is suggesting that there's only a tiny effect, if any, that the CBD has on serotonin levels. But I'm hopeful that it will become more clear once I finish collecting data. For future plans, I'm hoping to repeat my project using different concentrations to see if the amount of CBD affects the serotonin response. I'm also planning on running some tests with NHAS to determine what's inside of the CBD I'm using. This would help me determine if it's something inside the CBD that's having the effect on the worms, or if it's the CBD itself. I'd like to acknowledge NHAS, specifically Peter Feletra, Kelly Salmon, and Allison Michael, for providing me guidance and equipment throughout my project. Thank you so much for watching. I hope you enjoyed.
Okay, you got your standard issue fridge here, milk, yogurt, some greens, and a couple hundred worm plates.
Hello everybody, I'm Kai, and for my NHAS project, I tested the water quality of the Connecticut River and how that water quality is affected by the Wilder Dam. And I've always been very interested on exactly what the Wilder Dam does for the river. So I decided to do some tests on that. And I got a bunch of really cool science stuff. And I got to, boxes of Chemets tests, which are very cool. They're like these little test tubes that you snap. I got a microscope, which was a fantastic experience.
I got to test for E. coli and coliform bacteria, which was a very, very enriching experience. That was fantastic as well. And I also got to test for total dissolved solids with a super cool little TDS meter. Now, all this opportunity is provided by NHAS. It was so great. And I got to spend a lot of time outside. And it was just a really, really fun experiment. And I ended up learning a lot from it, too, because I found out that the water quality around the dam is actually worse. That trend continues down into the Connecticut River after the dam, which I thought was really interesting. And I'll definitely come back next year to either continue my research or to start another project. I want to thank everyone at NHAS for giving me this fantastic opportunity. It was a really enjoyable experience, and it was just overall a totally a great thing that I got to do.
Hello everyone, my name is Liam Markey. I'm a rising sophomore at St. John's Academy. So my project was testing for genetic diversity of showy lady slipper orchids in a fen in Stratford, Vermont. Now this research is important because showy lady slipper orchids are in decline throughout their range, and more specifically, they're critically imperiled in New Hampshire and vulnerable in Vermont. So I am testing 12 samples, 4 clusters within the fen, 3 samples in each, and ultimately my goal is to use DNA sequencing and alignment software to determine how similar each plant is to another. So just for a quick overview of the pieces of equipment I'm using in the experiment, this is a vortex. You use it to shake samples. As you can see, this is a centrifuge. You open it like this. And if I unscrew the safety cap, you can see where the samples go. So first in this process is DNA extraction. So first, you have to crush the sample with a micropestle. Then various buffers are added to remove contaminants and break cell membranes. Then the whole thing is washed through a purification column. And finally, it is eluted, and your finished product resides in an elution buffer. To amplify the region of DNA that you aim to sequence, you must perform PCR, or polymerase chain reaction. Now, PCR is performed in a very small 0.2 ml tube, which is then inserted into a thermocycler. This is a Mini 8. We are using these. They are small, compact, and fitting for a home lab that's only this big. So the Mini PCR thermocycler is controlled through a Mini PCR app. When it's connected to the computer, it can set cycles, temperatures, and it can show you DNA copies and where the thermocycler is at in the program. So to visualize the results of the PCR reaction, to see if it worked, we use gel electrophoresis. Now this is run on a small machine. We're using small machines called blue gels, and you take off the cap and turn this on, and it's a UV light transilluminator which can be used to visualize the results. So once you've confirmed that your PCR worked with gel electrophoresis, you must purify the PCR product and remove it of contaminants. This process is very similar to the second half of DNA extraction, so I won't go into it. But once you've purified your PCR product, you can't really sequence DNA at a home lab, so we send them off for sequencing to a lab in California called Eden Biosciences. My name is Liam Markey, and this is my home lab.
Hello, my name is Sarah Hall, and I attended the 3-week summer session at NHAS. My project was that I was trying to build an experimental platform for forming a multispectral image, and also trying to find a way to bring a cheaper multispectral camera to NHAS. I mainly chose to do this project because I wanted to gain more experience with more hands-on research, and also I never really learned about optics before. Basically, what a multispectral image is that it splits up the wavelengths of an image into more than its red, green, and blue information. This way, more information about the image can be extracted. To do this, I set up a camera and then I individually placed color filters in front of the lens to take many different images of the same setting, all with different color information. The thing that I was taking pictures of was a row of LEDs, and this way I could distinctly tell which wavelengths are being highlighted with certain filters, and this could be later used in the data processing. After I took the images, I transformed them all into grayscale so that I could have an easier time combining the images for the data processing. After that, I created a table which mapped out which of the LEDs seemed to be lighter or darker in all the different filter images. This way, I could find ways to subtract and multiply these grayscale images to highlight certain LEDs and also isolate them. I did this all on the image editing app ImageJ. After I did this, I put LUTs, or replaced all the grayscale values in the image with new fake color or pseudo-color values. This way, I could present the information in a different way, which made the LEDs pop out even more. My end result was an experimental platform where multispectral images were created that could distinguish between different LEDs. This is also the first step in a long process for introducing multispectral imagery to NHAS. In the future, I would definitely want to work with Fusion 360 to create a more compact camera, and also perhaps look into a neural network for the data processing. I'd also like to look into more uses for multispectral imagery in the NHAS lab.
With the COVID-19 pandemic causing most town swimming pools to be closed for the season, people are turning to naturally occurring swimming holes for recreation. Little is known about the water quality at some of these locations, particularly in downtown Lebanon, New Hampshire. Analysis of the water for coliform and other contaminants would help residents make safe choices about where to swim. Each location was visited for water sampling every 48 hours over a period of 2 weeks for a total of 6 samples per location. Water samples were taken in the morning and tested on site. Samples were also collected for a coliform analysis at my lab. The aim was to collect and analyze water samples over a period of 2 weeks to determine water quality and compare the relative safety of 2 common swimming areas in Lebanon, New Hampshire. Dissolved solid levels were very low and safe. The temperature at Great Brook fluctuated more because it was a smaller body of water, and it's also more shaded. For coliform, neither location was ever close to the high amount that is considered dangerous, but Mascoma had consistently higher levels. In conclusion, Great Brook has better water quality than the Mascoma River. Both sites have safe water for swimming and exploring. Mascoma had consistently higher coliform levels than Great Brook. However, these levels were not dangerous or unsafe. When I compared the data to the unsafe and normal levels of all of the results, what I got seemed to be safe. Both areas are suitable for swimming. I would personally swim at Great Brook. Mascoma has way more visitors, including humans and dogs, which may account for the increased coliform bacteria. My future plans include continuing to sample and test water over time. Also, I would like to share the results with the Valley News, and I would consider combining this project with my Daphnia research project at Lebanon High School. A huge thank you to everyone who helped me with this project, and thank you for watching!
Hi, my name is Miriam Wiesmensky. I'm a rising senior in high school. This summer, I focused my research on comparing the binding interfaces of SARS-CoV-2 and SARS-CoV with the ACE2 receptor. Given the pandemic we face today, researchers are racing to understand SARS-CoV-2, the virus responsible for the COVID-19 disease. Since the structure of the 2003 SARS virus is very similar to the present virus, I sought to compare how the viruses bind to the receptor that allows them to enter human cells. The binding interaction occurs between two proteins whose structures I could find in the Protein Data Bank, a publicly available resource. I spent the first week of my research troubleshooting and identifying the best modeling tools for my project. I visualized my proteins, compared their primary and 3D structures, and modeled their binding. Using the sequence and interface data I gathered, I then began analyzing and sorting my data in a spreadsheet. This was a hefty task, but yielded useful information. Finally, I prepared a presentation to share my findings. I was able to visualize the interactions across the interface between the viruses and the ACE2 receptor by mapping the types of interactions onto the sequence alignment using a color coding system. Understanding the similarities between these two viruses may aid in the development of therapies that target the ACE2 interface. After the session concluded, a few of us in the protein modeling group met to put together an instructional video about the resources we found most helpful. If you would like to learn more, visit the NHAS YouTube channel.
Hi, my name is Gabby Anzalone, and I did my project on comparing the effects of CBD and CBD with other compounds on C. elegans. This is where I did the last few rounds of my experiment. Before this, I was working in the upstairs of my apartment with no air conditioning, so this is much better. So to start my experiment, I had two different strains of C. elegans, MPR19, which is lacking cannabinoid receptor, and regular wild-type worms, which are typically used for scientific experiments. I tested 4 substances on the 2 strains of C. elegans and monitored them over 3 days. The 4 substances I tested were CBD and MCT oil, CBD and hemp oil, and just plain hemp oil and MCT oil as controls. So I started off with 5 L4 worms on each plate with a substance. Then after 24 hours, I moved them to a new plate with no substance and counted if there were any eggs or L1s on the plate, which I did 3 more times after 24 hours. Like I said before, I continued to monitor them for 3 days after transferring them to a new plate. I repeated this experiment 3 times. However, the results from the first 2 rounds ended up being trial runs due to some error. But on the 3rd try, I ended up working out all the details and the results were successfully collected. These pictures are of my data. They're pretty confusing and are still open to interpretation, but horizontally is the plate's data after 3 days, and vertically is the plate after it has been transferred to new plate after 24 hours. So what does all this data mean? Well, right now, I'm not quite sure. In my past experiments, it seems that all of the plates had relatively the same amount of eggs and L1s, which indicates that CBD does not have a big effect on the C. elegans. But for now, I cannot make a firm conclusion from my data.
So, what are we doing?
Transferring worms.
Nice. And what time is it?
10 o'clock? We'll be at this for another hour, probably.
Oh boy. Worms don't sleep, do they?
No, they do not, but neither do I, so it's okay.
So is this you picking up
Hi, my name is Sai Patel, and my project was on the comparison of the initial spike protein binding during viral entry of MERS-CoV, SARS-CoV, and SARS-CoV-2. Our world has experienced 3 major epidemics or pandemics caused by the coronavirus family over the last 2 decades. SARS-CoV-1, first discovered in 2002, MERS-CoV in 2012, and now the novel coronavirus, SARS-CoV-2, in late 2019. All of these viruses still have no effective vaccines or antiviral drugs to prevent or treat human coronavirus infections. Now, the virus's spike protein is what makes this equation so interesting. Apart from making the virus look spiky, is the protein that the virus uses to bind to a receptor. So if you can prevent attachment and fusion in the first place, you'll prevent entry, which makes it the ideal target for vaccine and antiviral research efforts. The overarching goal of my project was to better understand the residue interactions between the viruses and their receptors. By computationally binding all of the viruses to their receptors, I would be able to find the most probable mode of binding. The Protein Data Bank, or the PDB, is where I obtain protein sequences and structures to input into programs later on. I also input the PDB IDs of these proteins into UCSF Chimera to visually display it on my screen and interact with the structures in 3D. I aligned the sequences of my structures using Clustable Omega, a multiple sequence alignment program. I utilized the Sublime Text application to manually change certain parts of the protein chains so that my future applications would be able to run effectively. I used Haddock to dock my proteins so that I would be able to observe where interactions were and quantify how many interactions between amino acids and residues there were. Lastly, I used my results from Haddock to find the binding tightness, charge, intermolecular interactions, Gibbs free energy of binding, and polarity from these alignments in Prodigy. I'm still getting all of my results back, but hopefully my research can add to the puzzle of what exactly about the receptors and what exactly about those 3 viruses gives them their selectivity.
Hello, this is Alex, and I built a spectrometer at home. So the very first thing I did was I did a sketch of what my thing would look like. So from the left you have the light source, and then you have the slit right here that's restricting the amount of light through. And then you have the diffraction grating over here, which is splitting the light coming through the slit into the spectrum. And then you have the lens, which is the next line over here, which is focusing the spectrum onto points on the sensor, which is this box, and the lens and the sensor make up the camera. And then you can see my prototypes for the slit through here from left to right as they get better. The Lego one let too much light through, so I had to ditch that one. The cardboard one was much too big, and the slit was also too big. So I had to ditch that one as well. And then I got to the metal one here with the razors to block the light coming through. And I got the size of the slip using a piece of printer paper that I put in the middle. Then you can see my full sets up here. On the left you can't see my slit, but I built this one and then went from Legos to the cardboard, which I thought would be more practical because the Legos were really hard to keep on a level. And then I went to cardboard, and you see the setup, and then here I was taking an image, which I'm about to show you, and the slits in the back, which I was not currently using. Then you have, these are the 2 images I took without the slit. You can see the spectrum of light as it goes from blue to red. On this one, it starts as purple because I'm using the ultraviolet light. Then here is a more accurate thing of what I'm trying to get. I took this with the metal slit, and I used a fluorescent light bulb at my house. So this is the kind of reading I'm hoping to get from my spectrometer as it continues and as I finish it.
Hello, my name is Leanne Snow, and I'm a rising junior in high school. This year at NHAS, I chose to conduct a computational analysis to provide more information on receptors SARS-CoV-2 could be binding with in the human body. ACE2 is a receptor in the body that can be found in the vascular system. Among several other vital functions in the body, ACE2 assists in regulating blood pressure by participating in the renin-angiotensin-aldosterone system. ACE2 is also known to bind with several viruses such as SARS-CoV and SARS-CoV-2 to grant host cell entry. When bound together, SARS-CoV-2 appears to block ACE2's ability to relay information to biochemical functions in the body, potentially causing life-threatening conditions such as stroke. DC sign is another receptor in the body and is found on dendritic cells. It assists the immune system by identifying and binding to foreign bacteria. Research suggests that DC sign also binds with viruses to grant host cell access, including with the viruses SARS-CoV and SARS-CoV-2. Unfortunately, there is a dearth of information on how the virus interacts with DC sign, as the majority of information gathered on SARS-CoV-2 involves its interaction with ACE2. In theory, however, if DC sign and SARS-CoV-2 successfully bind together, DC sign may be unable to recognize and bind with pathogens. To investigate the binding potential of DC sign with SARS-CoV-2 versus ACE2 with SARS-CoV-2, I conducted several analyses using a variety of servers. I first conducted a sequence and tertiary structure comparison of DC sign and ACE2. The image to the left is a digital model of ACE2 and DC sign bound with SARS-CoV-2. The red and gray proteins at the bottom of the image are SARS-CoV-2. The gray at the top of the image is ACE2, and the darker blue protein is DC sign. And as you can see, ACE2 and DC sign do not align very well. The image on the right is a sequence comparison. The asterisks identify identical amino acids, which are the different colored letters, and there are not many pair of asterisks, as you can see, which concludes that the receptors are not similar to each other. Next, I compared ACE2 and DC sign's individual binding affinities, gives free energy rate, and interfacial contacts when bound with SARS-CoV-2 at 25, 37, and 40 degrees Celsius. The results suggested that DC sign and SARS-CoV-2 bind consistently stronger than ACE2 when bound with SARS-CoV-2. DC sign and SARS-CoV-2's Gibbs-free energy rate and binding affinity amounts were lower, meaning that their bonds were stronger. There were also significantly more interfacial contacts involved in their binding. As my study suggests that DC sign binds strongly with SARS-CoV-2, I believe that it is important to consider all the potentially dangerous outcomes this could have on the body. I hope that my research encourages other scientists to explore avenues that could offer more options for potential treatment for SARS-CoV-2, especially since there is a possibility that there are more receptors in the body SARS-CoV-2 could be binding with, especially since it binds with the receptors ACE2 and DC sign, which, as my study suggests, are quite structurally unique. I would like to thank the New Hampshire Academy of Science for providing me with mentors and equipment to conduct my analyses with, and thank you for watching.
Hi, I'm Annika Eastman. I'm a rising senior at the Sharon Academy, and for this program, I decided to do a molecular biology project on genetically modified organisms and their detection limit through PCR and gel electrophoresis. And for my experiments, I used a confirmed GMO soy and corn sample that had been sourced from a conventional farm near Indianapolis. And then I got 2 corn varieties and 1 soy variety from Sweetland Farm in Norwich, Vermont, that were thought to be GMO-free but needed testing to confirm this. And then for my PCR experiments, I used 4 different primers. I had a chloroplast and actin primer that served as my controls because those should show up in any plant. And then I used 2 primers for a promoter and terminator sequence from the cauliflower mosaic virus that is commonly used when making GMOs. And if I get bans for these primers, then it means that my corn or soy sample does contain GMOs. And the first of my tests was actually to confirm if my different soy and corn varieties contain GMOs or not. And for the samples that I thought were going to be GMO free, my first test actually showed that they did have evidence of containing GMOs, despite lab testing that had shown that they were GMO free. And so I had to do some troubleshooting and water tests and control tests, and it looked like my water samples potentially kept getting contaminated, and so I had to essentially start over and get new waters and primers. And finally, I was able to start testing the different percentages of GMO content to see if it was detectable. And to do this, I mixed leaf samples of non-GMO corn or soy with GMO corn or soy, and then I extracted DNA from these combined leaf samples. And ultimately, my testing showed that GMOs have evidence of being detectable at 25% and 10%, but I still need to do confirmation testing for some of the smaller percentages. I ran a 5% test that showed only slight evidence of containing GMOs, and then I did another water control test that showed I potentially had some contamination issues once again. And my hypothesis for this project was that the GMOs would be detectable below a 5% concentration. And this is the labeling cutoff for the legislation in the United States. And so I wanted to see if I could detect GMOs below that. And at this point in time, it's hard to tell if that is true or not. I've started to go into the lab to do some testing in a more controlled environment to see if I can get this contamination issue figured out, and so that I can repeat my experiments a little bit more accurately. And so far this project has definitely been a trial and error one, but I am of course very grateful to have had the opportunity to do this project and have the access to this equipment and be able to conduct our own research, and it's been a really great experience.
Hello, my name is James Eiler. For my research project, I conducted a conservational and thermodynamic study of DAS16 and FOXO. FOXO is a gene in humans which transcribes a protein, also called FOXO, which is critical to the insulin signaling pathway. DAS16 is the homologue of FOXO that is found in the C. elegans. DAS16 in the C. elegans transcribes a protein which is critical to the insulin-like signaling pathway, or ILS. There are several forms of FOXO produced by different genes. For DAS16, however, there are different isoforms. Isoforms are proteins produced by the same gene which have several small differences. DAS16 FOXO bind to promoters containing their binding domains and initiate transcription. In humans, FOXO initiates glycogenesis, which breaks glycogen into glucose, which raises blood sugar levels. Insulin in humans, or insulin-like peptides in C. elegans, bind to receptors on the outside of cell membranes and initiate a process that downregulates DAS16 FOXO. For my project, I wanted to determine how well DAS16 is conserved between species and how differences in amino acid sequence affect binding between DAS16 FOXO and their promoters. This would give us more information on how well conserved DAS16 FOXO is, in particular the various parts of DAS16 FOXO, such as the binding and non-binding areas. My hypothesis was that the binding domains would be conserved between different species, but that the non-binding domains would be less conserved, and that the binding affinities would differ between different forms of FOXO and isoforms of DAS16. To conduct my research, I compared the sequences of DAS16 in C. elegans and FOXO in humans, and also in 6 other species, to determine how conserved the amino acid sequence is between species. Further, I determined the binding affinities of 2 different isoforms of DAS16, isoforms A and H, and 2 different forms of FOXO, 1 and 3. I first conducted sequence alignments using a software called Cluster Omega, which compares the amino acid sequences and gives a value for similarity, which is called percent identity. Secondly, using a software called Rebetta, I generated the first 3D structures of a complete DAS16 protein. Then, using the software Chimera, I compared the 3D structures of DAS16 and FOXO to make sure that the binding domains were in the same area and that the structures were similar. I generated a total of 4 structures for DAS16, 2 for isoform A and 2 for isoform H, one each using FOXO1 and FOXO3 as a template for folding, and so I compared the structures of all of those to FOXO1 and FOXO3. Then I used the server Haddock to bind the proteins with their DNA binding domains using a process called docking. Then I put those docked structures into the server pre-DBA to calculate the Gibbs free energies of binding. The percent alignments between DAF16A and DAF16H in FOXO1 and FOXO3 range from 30 to 37%, indicating a very high degree of overall variance. However, the binding domains were very well conserved, with relatively little difference between them. This trend held true with the comparisons with the other species, specifically rats, bovines, pigs, zebrafish, African clawed frogs, and chickens, which all showed increased deviance in the non-binding areas and relatively little deviance in the binding domain. The lack of variance in the binding domain suggests that those areas were crucial to the function of the protein, and that the other areas are less crucial, and thus can diverge more between species. Further, the Gibbs free energies of binding were all the same, which contradicts my hypothesis. This suggests that the small differences between the binding domain amino acid sequences have no material impact on binding, which it further implies that DAF16s and FOXOs are somewhat interchangeable in function, and that they can serve the same purpose in humans and C. elegans. Further, the lack of difference between the binding affinities of isoforms suggests that differences of certain mutants are not the result of changes in the DAF16 gene, but rather possibly to changes in other parts of the insulin signaling pathway or insulin-like signaling pathway. Thank you for watching.
Hello, I'm Trace Barnhart, and using various protein modeling and analysis software, I analyzed the similarities and differences of the ACE2 protein in humans and dromedary camels. The ACE2 protein is important because it's the entryway into cells for the SARS-CoV-2 virus. Due to my literature review, I hypothesized that the ACE2 in dromedary camels would be unable to bind with SARS-CoV-2. My results, however, seem to completely contradict this hypothesis. Thermodynamic data, calculated by the software known as Prodigy, showed that not only could the camel ACE2 protein bind with SARS-CoV-2, it actually bound more effectively than with the ACE2 protein of a human. This turned me back to the literature, where further analysis showed the paper I had reviewed earlier was actually not backed up by any other papers, and only completed the first step of the analysis I performed, being sequence alignment. Furthermore, I found 7 separate papers, all stating that a sequence alignment is not enough to determine binding accurately. Additionally, my data showed that the part of the protein they said could not bind would actually bind 6 times, 2 more than the same part in the human protein. In the end, I concluded that this paper simply did not have solid enough evidence, and found that the camel ACE2 protein can indeed bind with the SARS-CoV-2 virus. A study performed on actual proteins would be the best way to confirm my results. So, this whole experience, I would say the worst part of the experiment was probably, working on the files in Sublime Text searching for errors was quite frustrating. However, I would say even more frustrating was the way scientists name things. I spent 4 or so hours trying to figure out what ACE2 binding had to do with RNA binding motifs, before figuring out, with help from Allison, that RBM also stands for receptor binding motifs, which is slightly different from the thing I was already working on, being RBD, which stands for receptor binding domain. Now, I would say the best part of this whole experience was learning that you can visualize and analyze the way fundamental molecules for life interact solely through use of software. That fact alone just blows me away. The technology can accurately simulate life? Wow.
Hello, my name is Sora Shirai, and my research compared various ACE2 receptors from different animals and looked at their binding with the SARS-CoV-2 spike protein. And so the ACE2 receptor, if you don't know, is the main receptor that's currently thought to be the entry point for SARS-CoV-2 in our cells. And I tested for 7 different animals, so 4 mammals, the cat, the mouse, the masked palm civet, and the Chinese rufous horseshoe bat, and also 3 non-mammals, the Chinese soft-shelled turtle, the zebrafish, and the chicken. And so for these different animals, I first found their PDB files, which are these files with all of their protein sequence and the protein structure. And so I found these files from RCSB Protein Databank and the Swiss Model Databank, and then after that I observed the protein structures using Chimera, and then I did protein sequence alignment and compared the similar amino acid residues using Cluster Omega, and then after that I used HADOC 2.4, ClusPro, and ZDoc to do protein-protein binding between the ACE2 receptors and the SARS-CoV-2 spike protein to create PDB files with the 2 proteins binded together. And after I got that, I took those PDB files and I put them into Prodigy and PDB PISA to get the binding affinities between the proteins and also to calculate the interacting residues. And so after I did that, I found that I can't really conclude anything because all of my work was computational, and so if binding really occurred between these 2 proteins in real life, there may be other factors that weren't included in these calculations that might occur, but based on the results that I got, I found that the masked palm civet actually had the highest binding affinity, and the zebrafish had the lowest binding affinity. Surprisingly, the cat was the second lowest, which is surprising also because cats have been found to have SARS-CoV-2. And then in terms of similarity with the human ACE2 receptor, based on protein sequence, I found that the cat was the most similar and that the zebrafish was the least similar. And then in terms of similarity with the human ACE2 receptor, based on the number of ACE2 residues that were interacting with SARS-CoV-2, the spike protein of SARS-CoV-2, in both ACE2s, the chicken had the most number of those residues and the horseshoe bat had the least number of those residues. And so that was my research. Thank you so much for listening, and thank you so much to the NHAS lab for making this opportunity available to me even during this pandemic.
Hello, my name is Luke Youngster, and over 3 weeks as part of the NHAS research program, I worked on adapting a micronucleus assay for C. elegans. The C. elegans organism offers many potential benefits to in vivo genotoxicity testing because it is relatively cheap and easy to access and allays many of the ethical concerns of using rodents. It, however, retains many of the benefits of using these organisms due to its conserved metabolic processes and DNA repair pathways. Micronucleus testing analyzes the damage done to an organism's genome by evaluating the frequency of micronucleated cells, which results from lagging chromosomes or parts of chromosomes not properly incorporated into daughter nuclei during cell division. This is commonly caused by DNA strand breaks, failure of chromosomes to separate, or damage done to cellular mechanisms involved in mitosis. The objective of my study was to expose C. elegans to known genotoxins, isolate and then stain their cells, and compare the micronucleated cell frequency with a negative control. Because micronuclear formation in C. elegans has not been thoroughly investigated, I want to use compounds known to cause other genotoxic responses in C. elegans, as well as induce micronuclei in rodents. For the initial study, I used vinblastine and doxorubicin, both chemotherapy drugs that induce genotoxic damage through different mechanisms. To expose the worms, I pipetted dilutions of these compounds as well as RO water as a negative control onto petri dishes and let these set into the agar. I then let C. elegans worms develop on these plates. To isolate the cells, I first dissolved the worms out of cuticles with tritone. I then used the proteinase papain as well as mechanical disruption using probe sonicator to further dissociate the cells. I then centrifuged the resulting cell suspensions onto cover slips fix them in methanol, and permeabilize them using Triton. I used several different staining methods during initial testing. First, I used a fluorescent Dabby stain, a methyl green stain, and then a methyl green with an eosin white counter stain. All were largely unsuccessful. In a second test using all negative controls, I successfully stained nuclei with DAPI using a slightly altered protocol, and plan to apply this procedure to worms exposed to positive control compounds in the future.
I'm running a validation on our brand new UV-Vis spectrophotometer. We'll use this to get Zia's measurements for copper and iron concentrations in the tailings of the Elizabeth Mine in Copperas Brook and Lord Brook.
Hi, I'm Zia, and I did my project on the effects of the Elizabeth Mine Superfund site on the water quality of the west branch of the Ompompanoosuc River. The Elizabeth Mine was discovered in 1793 and produced copperas, also known as iron 2 sulfate or ferrous sulfate, as well as copper. Eventually, due to lack of money and demand for copper, the mine shut down in 1958. When it shut down, sulfide ore was left exposed to oxygen and water, leading to acidic mine drainage from tailings piles and waste rock. These minerals were carried into the west branch of the Ompompanoosuc by Copperas Brook and Lord Brook. In 2001, the Elizabeth Mine was added to the Superfund list by the Environmental Protection Agency, with work beginning in 2003. The revitalization plan, which centered on isolating copper tailings, leach piles, and waste rock from waterways, was completed in 2010. So, my experiment will explore the effectiveness of the Superfund cleanup in the Ompompanoosuc River, the hypothesis being that the data collected on levels of dissolved oxygen, ammonia, phosphates, copper, iron, total dissolved solids, conductivity, salinity, and turbidity will show an improvement on the water quality when compared to data collected by the state of Vermont over the past 20 years. A study of the macroinvertebrates will provide information on the improved habitability of the water, as well as the recovery of the aquatic ecosystem as a whole. While out in the field, I looked at testing 9 sites. 3 of those sites were potentially bringing minerals down from the mine. Those 3 sites were Copperas Brook, Lord Brook, and what is believed to be a mine drainage site. I tested above and below these 3 sites on the Ompompanoosuc in order to understand their effects on the water quality of the river. I alternated testing between sites A and F, Copperas Brook and the mine drainage sites, and G through I, Lord Brook sites, every other day. I performed all water quality tests in the field except for the iron test and the analysis for the macroinvertebrates. For my results, I was never able to detect any ammonia or phosphates in the water. This is a good sign because excess ammonia and phosphate can be an indicator of fertilizer leaking into the waterway and can lead to early eutrophication. The turbidity was tested once at the beginning of the experiment and was determined to be less than 5 NTUs, which is very good because it means that the water is very clear and doesn't have excess particles polluting it. Overall, the levels of dissolved oxygen, pH, conductivity, total dissolved solids, and salinity were fairly similar throughout the Ompompanoosuc test locations. This is encouraging because the high levels from Copperas Brook and the drainage site appear to be not affecting the larger river. While no copper was detected at Copperas Brook, there was a small amount detected at the drainage site, with a mean of 0.36 repeating parts per billion. The drainage site also had a very low pH, averaging 5.75. Initial results from the iron testing also indicate iron levels well above the chronic level of 499 micrograms per milliliter. After analyzing the macroinvertebrates, all Ompompanoosuc test sites and the Lord Brook site have a range of different families of macroinvertebrates, including the mayfly and stonefly orders, who have a low tolerance for pollution. However, the Copperas Brook and drainage sites have no mayflies or stoneflies, indicating it is not habitable for those species. While the Ompompanoosuc itself shows encouraging statistics for recovery for the ecosystem and water quality, and Copperas Brook appears to be doing better than past data indicates, the drainage site appears to have slightly acidic and mineral-rich water, which could lead to the problems for the rest of the river if not dealt with carefully.
Hi, my name is Leanna Kish, and I'll be going into my senior year at St. Paul's School, and I did my research on the effects of Joule e-liquid and its components on the behavior of C. elegans. So to do my project, I used a chemotaxis experiment. So I was tracking the movement of the worms. And to do that, I drew a bullseye on the bottom of my worm plates. And the middle section was considered section 1. The next circle was section 2. The outer circle was section 3. And then the walls of the plate I called section 4 because the worms began to crawl up the sides of the plate, and at first I thought they were disappearing, but they were just trying to escape the plate. So those are my 4 sections. I would put the solution that I was testing in the center of the plate, so in that middle section, and then I would transfer 10 worms into that solution for every plate. So for each solution, I had 4 concentrations, 1%, 25%, 50%, and 100%. And I had 2 test plates per concentration, each with 10 worms. So 20 worms total per concentration, 4 concentrations per solution. The solutions that I tested were Jewel menthol, that's the original Jewel product, and then I tested the individual ingredients that are found in a Jewel pod, which are benzoic acid, nicotine, glycerol, propylene glycol, and menthol flavoring. Now, Jewel doesn't explicitly say what they use to make their menthol flavoring, so we just used straight menthol. And benzoic acid and nicotine are both not very soluble in water, so I made those stock solutions with propylene glycol, since that is what Jewel is majorly made up of. There's 5% nicotine solution in a Jewel pod and 4.5% benzoic acid, and then the propylene glycol and glycerol are in a 7:3 ratio. So I replicated those with the solutions that I tested. I also made a mock Jewel solution where I put all of the ingredients together in the recipe that Jewel supposedly uses, and I tested that as well. I have not yet crunched the numbers, but my little assistant here is going to be helping me do that very soon. So I'm excited to find out what happened.
Let's have a round of applause for all of our fantastic summer researchers. We are so proud of all the amazing work you've done this summer.
And we'll see you next summer, hopefully in the lab.